polycistronic mRNA, what that means is it can include multiple genes for translation in a single mRNA. And as I mentioned before, when you transcribe DNA, the mRNA that results will be complementary to the coding strand, which means it will look almost exactly like the template strand, with the exception of uracils in the place of thymines. And this mRNA, once transcribed, is sent to a ribosome where it is translated into protein. Then we come to tRNA, which is a key part of translation. tRNA is synthesized by RNA polymerase III, again, only in eukaryotes because prokaryotes have a single RNA polymerase, which we will talk about later. This tRNA functions in basically translating the language of RNA into amino acids, which are incorporated into proteins. How it does that is it binds to mRNA at a ribosome using these three nucleotides here, which is called the anticodon. And this is anti-parallel to the codon present in mRNA so that it can bind properly and only bring the appropriate amino acid. The amino acid will be attached at the tail end of the tRNA at this CCA region, and it is activated by amino acyl tRNA synthetase, which binds each tRNA to its specific amino acid. Then finally, we have rRNA. This rRNA, it has a very important function in ribosomes where it forms over 60%. And it is synthesized in eukaryotes by RNA polymerases 1 and 3 at the nucleoli. This rRNA also has enzyme function, which is called a ribozyme in this case. And what that means is it can splice its own transcripts, which we'll talk about later. And it can also, that's why it has such an important function in translation. And this rRNA, as part of the ribosome, creates the perfect location and energy for translation to occur properly. So now we've come to a couple practice questions that I have for you guys over the review. I'll give you a couple minutes for each question and feel free to type the answer in chat once you know. Great. It seems like a lot of you guys have the answer already, and that's B, ionic bonds. Does anyone mind explaining how they got to that answer? If not, I can just go ahead and explain. So in DNA, we have the sugar phosphate backbone, which automatically eliminates. Yes, but hydrogen bonds, Michael, yes, DNA does have hydrogen bonds between the base pairs, but these hydrogen bonds aren't necessarily covalent. The covalent bonds actually come in between the phosphate and sugar backbone. So that's why we can also knock out C along with the sugar and the phosphate group both appearing in DNA, the covalent bond formed between the sugar phosphate groups. Yes, not a problem. So I have another question for you guys. Actually, this one is A, and I'll explain why. Promoters actually exist in both prokaryotes and eukaryotes because they are the parts of the DNA that actually allow binding of RNA polymerase so that transcription can occur. Yes, I can explain PribNow boxes. And the PribNow box is actually a part of the DNA that binds to RNA polymerase, and it helps identify the location for the transcription start site. It is generally located around 20 to 40 base pairs upstream of the transcription start site. But this TATA boxes or TATA boxes and PribNow boxes are the respective regions in the promoters for eukaryotes being TATA and prokaryotes being the PribNow box, as where RNA polymerase actually binds to the DNA to start scanning along the DNA for the transcription start site. Does that make sense? Um, for Michael's question, you don't necessarily have to have introns for genetic diversity. To the best of my knowledge, I can say that this is because of bacterial methods like transduction, conjugation, and I forget the last one, which explains a lot of why bacteria can transfer material to each other and learn a lot about the environment from each other. Yes, transformation. Does that answer your question, Michael?
Yes, thank you.
Yeah, not a problem. And Sarah, if bacteria do not have introns, is that why they are polycistronic? To be completely honest, I cannot say for sure. I believe they are polycistronic because it's a more efficient process to transcribe everything because in bacteria, translation begins almost immediately after an RNA fragment is available. And that allows the transcription of many genes or translation of many genes at once, which contributes to their fast turnover and replication. So I'd have to give you a tentative yes, because you can't really avoid the sections in between when you're transcribing a gene. But I definitely don't know for sure, and I don't want to give you the wrong information. Sorry. And I have one final question for this section, but I do want to point out this is for a eukaryotic gene with the promoter and the introns in between, which are then spliced out later, as I'll review in a couple minutes. And here's the last question for this section. Again, feel free to type it in chat if you guys have an answer. So I'm getting a little bit of a mixed answer here between B and D. So I'll go over RNA polymerase in eukaryotes again, just to make sure that we're all clear on it. So RNA polymerase 1 is actually in the nucleolus, and it only synthesizes ribosomal RNA, or rRNA. For RNA polymerase 2, that is actually the right answer, and it only synthesizes some nuclear RNA and also mRNA, and it's located in the nucleus. RNA polymerase 3 is also located in the nucleus, but it synthesizes transfer RNA or tRNA and some ribosomal RNA. So RNA polymerase 1 and 3 would actually end up being the right answer for the synthesis of ribosomal RNA, since they both have that ability. I'm gonna keep moving forward now. So now we are at transcription, which is the actual process of changing the DNA language into RNA language. So Sim just mentioned a mnemonic, which she uses to remember which RNA polymerase corresponds to which RNA product. And she said it was 123 RMT, which is honestly something I've never heard, but it works and I will remember it as well. So I hope it works great for you guys as well. But back to transcription. Transcription is the process of changing the DNA into RNA so that it can later be converted into a gene product. So I just want to review the machinery just so we can understand what the steps include later. So first we have DNA helicase and DNA topoisomerase. What DNA helicase does is at the location of the DNA, where the gene is that we want to transcribe, DNA helicase actually separates the strands. And as it does that, as it straightens out the area and opens it up for transcription, the areas further ahead are going to end up being coiled tighter. And you'll notice that if you have like two strings and they're wound with each other and you try to separate them, you'll notice that the areas further ahead and further behind are going to end up winding tighter. And to prevent that, we have DNA topoisomerase, which actually relaxes the supercoiling so that transcription doesn't suddenly stop. And we have an open area for transcription. And helicase and topoisomerase are both enzymes. Then we have, for eukaryotes, we have RNA polymerase II, which binds the TATA box. And once it binds that box and it starts moving down and recognizes the transcription start site, or TSS, it continues just reading the coding strand or the antisense strand from 3' to 5' and adding nucleotides, RNA nucleotides that are just available in the nucleus to the RNA transcript. Now, on the other hand, in prokaryotes, as I've been mentioning, there is only one RNA polymerase, but it is also DNA-dependent RNA polymerase. And the way that it differentiates between different sets of genes is by using a regulatory sequence called a sigma factor. And there are many sigma factors available in prokaryotic cells that differentiate between a lot of different sets of genes. Now, both RNA polymerases for prokaryotes and eukaryotes, they don't use a primer or perform proofreading. Once they bind the TATA box for the eukaryotes and the PribNow box for prokaryotes, they just start reading and adding nucleotides. To help with this process, to help RNA polymerase find and bind the correct area, the promoter, there are transcription factors, which we will explore in further detail when we get to enhancers. And what they do is they just help increase the transcription by leading RNA polymerase to the right spot for binding. For termination of translation, we have two different pathways, row-dependent and row-independent termination. So in bacteria or prokaryotes, there is a factor called Rho, which binds to the RNA transcript behind RNA polymerase and starts moving along it as RNA polymerase is transcribing the RNA. It helps induce termination of transcription when it catches up with RNA polymerase, which can happen when RNA polymerase includes an area of rich in G's and C's, guanines and cytosines, and their stronger bonds help form a hairpin loop, which forces RNA polymerase to stop and Rho to catch up. And when Rho catches up, it forces RNA polymerase to stop transcribing. Another way, in row-independent termination of transcription, what happens is as RNA polymerase is transcribing, it reaches an area rich in A's and T's, and the weak bonds between A's and T's naturally disassociate RNA polymerase from DNA, causing it to end transcription. So the primary transcript in transcription is HN RNA, or heterogeneous nuclear RNA. And this is the primary transcript. Yes, Michael. How can you tell the difference between antisense and sense and template and non-template? Let me go back one slide. So the way that it's been named, the coding strand is the strand that RNA polymerase actually binds to, because that's what's used to code the mRNA. However, the coding strand is also known as the antisense strand because it is the exact complement to the RNA that actually makes sense when translated. If you look at the above picture, you will notice that the RNA transcript and the sense strand are extremely similar, with the exception of U's in RNA in the place of T's in the DNA. And that's why the strand that isn't bound to RNA polymerase is called the sense strand, because it actually would make sense if it were converted, if it were used in the place of T's. Does that make sense?
Yeah, but my question is, how do you look at it like by a certain sequence? Like if you had two, since everything looks the same, like do you just go by the top strand? Is that the coding strand, or you know what I mean? Like how would you tell? Like, if you didn't have this directionality laid out.
You'd have to look at the RNA transcript. Without the RNA transcript, you can't tell. And there's usually genes that can be transcribed in both directions in DNA. So until we look at a specific RNA transcript or mRNA, there really is no antisense or sense strand.
Okay, cool. Thank you.
Yeah, no problem. So then we've come to hRNA, which is considered pre-mRNA because there's a lot of changes that need to be done to hRNA to create the actual mRNA that is then translated. The hRNA includes the introns that were in the DNA, along with a 5' and 3' untranslated region, which are still there for specific reasons, like the existence of the start codon, the ATG or the AUG that you see in the picture. That comes at the end of the 5' untranslated region. So that explains its inclusion. And the 3' untranslated region includes a poly adenine addition signal, which shows up as AAU-AAA in RNA. And what that does is it codes for the addition of a lot of adenines to the end of RNA during post-transcriptional processing, so that RNA doesn't get degraded after transcription. Because as soon as RNA is released from transcription, there will be enzymes that try to break it down from both ends. And that polyadenal addition signal is what prevents that for long enough to allow translation. So then we've come to processing, and I just mentioned a portion earlier with the poly A tail that prevents degradation of the RNA in the time that it takes to be translated. What it also does is it aids transport of the mRNA out of the nucleus into the cytoplasm, where it can bind to ribosomes. Additional processing is also done with introns, where they're spliced out using small nuclear RNA and SNRPs. What they do is they bind to the 5' and the 3' ends of splice sites at the introns ends, and they cut the introns out, ligate the exons together, and force the introns to form a lariat shape, which basically looks like a lasso, where it will bind to itself, and that signals it to be degraded in the nucleus later. An additional processing is a 5' cap added on after RNA transcription starts. And what this does is it also prevents mRNA degradation, but from the 5' end, just like the poly A tail does at the 3' end. We've mentioned before how bacterial transcripts are polycistronic, which means they can have many protein products coded from one mRNA. What eukaryotic cells have similar to that is alternative splicing. And what that means is for each protein product, for most protein products, you may be able to use the same RNA, but splice out a different set of introns, leading to a different mRNA because of the exons and introns that remain in the coding region. And that allows for a different protein many different proteins within the same hnRNA. And please ask questions if you have any. After all this processing, the mRNA still includes the untranslated regions because of the signals and the prevention of degradation. And it exits through the nuclear pores and it attaches to the ribosomes in the cytoplasm. So I have another set of questions for you guys at the end of the transcription section. So please just type the answer in chat or say it out loud if you have it. So it seems like everyone's got the right answer for this one. And does everyone understand what the prevention of supercoiling actually does in aiding transcription? I guess I'll take that as not necessarily prevents incorrect pairing, but it keeps the DNA, it allows the DNA to be open enough so that transcription can continue and the RNA polymerase doesn't get stuck as the DNA is wound ahead of it. It allows helicase to separate the strands without excessive tension because DNA topoisomerase relieves the tension by uncoiling DNA ahead of the transcription area. And let me know if that doesn't make sense and I can explain it further. So for the next question, what does the 5' untranslated region end with? Great, it seems like I've been getting B's as answers and that's correct. The 5' untranslated region ends with the start codon and it starts with the transcription start site. The since the untranslated region occurs from after transcription, since it's been transcribed and isn't going to be translated, it doesn't include the promoter, because the promoter is actually the section in the DNA that allows transcription, which occurs after the promoter or downstream of the promoter in the gene. It isn't an intron because it's bordered by the transcription start site at one end and the start codon at the other. And after the start codon is immediately an exon. So it's not near an intron. And let me know if that doesn't make sense. And this can be seen here with the promoter upstream and the 5' UTR being bordered by the transcription start site and the start codon. The TSS is the transcription start site, which is the signal in DNA that tells RNA polymerase II, in eukaryotes at least, please start transcribing here. Does that make sense? Great. And then a final question. So this is something I wanted to clear up, because the first time I learned about it, I was very surprised, having taken bio for many years and not having known this. But the mRNA actually attaches to ribosomes in the cytoplasm. And while we may learn about it in like middle school that, oh, the rough ER is where ribosomes are, mRNA actually attaches to ribosomes in the cytoplasm, but any signals within the mRNA will tell the ribosome to move to a specific place. And if the protein is going to be secreted for movement throughout the body, or if it's going to be on the cell membrane, so it needs to be processed by the Golgi body, it will signal the ribosome to move to the ER, so it can be more easily processed through the ER and the Golgi body for movement. Does that make sense? Great. So now we are going to move on to translation. Now this is the, what's an example of a question that asks about the ER? As in like on the MCAT? Oh, I'm not sure what you mean, Lenore, if I'm saying your name correctly. Do you mind explaining?
Hi.
Hi.
So basically what I'm saying is, since mRNA attaches to ribosomes in the cytoplasm, sometimes we get confused whether it's asking about, okay, smooth ER, rough ER. So what would be, like, how would the question address that? Is it just mRNA, okay, it's in the cytoplasm versus, proteins are made in terms of the rough ER, something to that effect?
Let's see. Well, proteins aren't necessarily made at the ER. It's only if they're going to be processed by the ER later and need to be sent to the Golgi. That's the only time where they are made at the ER. And that's why we have the rough ER, because so many of proteins do need to be processed to be sent somewhere else. If that helps answer your question.
Yes, thank you.
Okay, great. One second. Okay, so for translation, we have as the main location of translation the ribosome, which is made by ribosomal RNA and proteins. Oh, and just before I move on to explaining more about ribosomes, I do want to mention that for translation to occur, we need the mRNA transcript, which serves to provide the instructions for which amino acids are in the protein. We need the tRNA to actually bring the amino acid. We need the ribosomes to provide a location and a favorable binding conformation. We need the actual amino acids to form the protein, and we need GTP to assist in providing the energy for forming separate bonds in each of the steps. So for the ribosomes, like I mentioned before, we have ribosomal RNA and we have proteins. And the ribosomal RNA for the ribosome is transcribed by RNA polymerase 1 and RNA polymerase 3. Now, this RNA is split up between two different subunits, the large and small subunits, which does happen in both prokaryotes and eukaryotes. And these subunits bind to each other only during the translation process. What the ribosome does is it reads and it holds the mRNA at each of its specific sites, which we'll get to later. And that allows the proper tRNA to bind and bring the proper amino acid. Now, there are differences between the eukaryotic and prokaryotic ribosomes. And I just want to tell you guys ahead of time that don't worry about what the S means. That's just for the sedimentation of the parts in the ribosomes. And it's just used as a number to describe the different strands that are inside the ribosomes. So the eukaryotic ribosome is considered an 80S ribosome as a whole, and it includes 4 strands of RNA, and the rest are proteins. 3 of these strands, the 28S, 18S, and 5.8S, are actually transcribed in the nucleus together as one set of genes, and then they're processed and cut down to the 40 to a 40S transcript when they split into the 28S, 18S, and 5.8S. However, the 5S portion is transcribed separately by RNA polymerase 3 outside the nucleolus. And these 4 different strands of RNA come together to form a 40S smaller subunit, which includes the 18S RNA and proteins, and the 60S large subunits, which includes the 28S, 5.8S, and 5S RNAs with proteins. On the other hand, the prokaryotic ribosome only has 3 strands, and it's a lot more straightforward in that the 30S subunit just includes the 16S rRNA and proteins, and the 50S subunit, large subunit, includes the 23S and 5S strands and proteins. I know this is a lot of information about like numbers and S values, but it would just help to remember that the eukaryotic one has more strands and it's transcribed separately. And the prokaryotic one is a 70S and it has less strands. Actually, the difference in between the eukaryotic and prokaryotic ribosomes is what allows drugs to target prokaryotes specifically when we're infected. So there are 3 steps to the actual process of translation, which are initiation, elongation, and termination. And we'll go with each of them in order. So for initiation, the first step is the small subunit actually binds the mRNA, and the location that this occurs varies based on if it's a eukaryote or a prokaryote. In prokaryotes, there's a sequence like the Pribnow box, but in mRNA, which is called the Shine-Dalgarno sequence. That's located in the 5' untranslated region of the mRNA. In eukaryotes, the small subunit just binds to the 5' cap that's inserted during post-translational processing. At this time, the first tRNA, called the initiator tRNA, also binds to the small subunit at the P site, which I will explain each site later. This initial initiator tRNA includes the amino acid methionine for eukaryotes and a really long name, but I'm just going to call it formylmethionine for prokaryotes. So they do include different different amino acids for the initiator tRNA for prokaryotes and eukaryotes. After the binding of the initiator tRNA at the P site, the small subunit scans along the mRNA downwards from 5' to 3', and once it finds the start signal, which is AUG in the case of both prokaryotes and eukaryotes, it will bind the large subunit. And this association is assisted by initiation factors that help the ribosome bind only during translation. They don't always stay with the ribosome. So I just wanted to compare eukaryotic and prokaryotic initiation one more time. And in eukaryotes, the ribosome binds the 5' cap and moves to the start codon. And the initiator tRNA includes the amino acid of methionine. On the other hand, in prokaryotes, the ribosome actually binds the Shine-Dalgarno sequence in the 5' untranslated region. And this sequence is in the untranslated region before the start codon. And the amino acid brought by the initiator tRNA is N-formylmethionine. And the Shine-Dalgarno sequence actually is what allows the prokaryotes to have polycistronic mRNA, because each Shine-Dalgarno sequence can signal the ribosome as to the location of a different protein or gene. So all the ribosome needs to do is scan for the Shine-Dalgarno sequence to translate a protein. So then we come to the next step of translation, which is elongation. And this step repeats as soon as initiation starts until translation is terminated. Well, now that the ribosome is bound with both subunits to the mRNA, it moves downstream from 5' to 3', and it adds tRNAs as it encounters new codons. So there are 3 sites within the ribosome for binding, and from the first site the mRNA encounters, which is the A site, I'd consider that the landing site. And what that does is it allows a new tRNA to bind to the mRNA with its complementary sequence. The P site, which is where the first initiator tRNA bonds with the start codon, is the site that holds the tRNA that has the polypeptide chain attached. And once an initiator tRNA is moved to the T site and a new tRNA is added to the A site, peptidyltransferase, an enzyme, is used to bind the amino acid from the tRNA in the P site to the amino acid in the tRNA on the A site. And then it moves forward one more step to the exit site. So the tRNA that was previously in the P site moves to the E site, and that's where at that point the tRNA is uncharged, which means it does not have the energy nor the amino acid, and it leaves the ribosome. Now this whole process is assisted by a set of elongation factors, and there's 3 of them: EFTU, which binds charged tRNA and releases it to the ribosome for binding in the A site, and that uses ATP hydrolysis for the energy for binding. Then the next elongation factor is EF-T, which removes the low energy GDP from EFTU and adds GTP to EFTU for another round of tRNA binding. Then there's the third elongation factor, which moves peptidyltransferase back from the A site to the P site for another round of peptide bond formation. And this peptidyltransferase is part of the large ribosomal subunit, and it uses GTP to create that bond. In addition, there are signal sequences, like I mentioned earlier, that cause ribosomal, as the mRNA is being translated, the ribosome recognizes these signal sequences and moves to the ER for secreted proteins. I know that was a lot of information, so please let me know if you need anything explained further. So the final step of translation is called termination. And what that does is it ends translation and releases the polypeptide for post-translational processing. How that begins is when a stop codon is recognized in the mRNA at the A site, a release factor comes to the ribosome and binds the stop codon, which induces the addition of water to the polypeptide. Now, transcription factors and peptidyltransferase recognize this water being bound to the polypeptide, and it helps them hydrolyze the polypeptide from the final tRNA and release it to where it needs to go for further processing. This uses ATP for the hydrolysis of the polypeptide from the tRNA, And it also induces the ribosomal subunits, the release factor, and the mRNA to disassociate. Finally, there's processing of the proteins, and there are many different ways that this can occur. A key way this occurs is by phosphorylation, which activates the protein by adding a phosphate group. It may also deactivate the protein, and this is done by protein kinases. The proteins are phosphorylated at the serine, threonine, or tyrosine residues, which all have open hydroxyl groups to accept the phosphorylation. Another way proteins can be modified is by carboxylation, and you'll see this when I exhibit prothrombin on the next slide. You can also glycosylate a protein and send it to the membrane to act as a cell membrane signal. Or you can add lipid groups in prenylation for other membrane-bound enzymes. You can lyse a protein to create its active form, and you'll see that with insulin. You can also see quaternary structure form with translated proteins where several different polypeptides come together to form a product such as hemoglobin, which includes 4 different polypeptides. A final step that proteins do go through after translation is folding. And there are chaperone proteins available, which give the protein a safe environment to fold in, which is why I assume the name is called chaperonin. And what they do is they allow the folding process to be faster while preventing the wrong portions of proteins from sticking together. And they also shield the proteins from folding to prevent any unnecessary bonding within itself or with other proteins. And I just wanted to show you guys the different modifications that can be done to proteins. And so the example of prothrombin is a clotting factor, and to be an active clotting factor, it requires the carboxylation of its glutamic acid residues for proper function. And this modification actually requires vitamin K, which is why a lack of vitamin K can actually cause excessive bleeding or bleeding disorder. So now that we've finished with translation, I do have another set of questions for you guys. Like always, feel free to type it in chat or say it if you have the answer. Great. It seems like everyone has a really good grip on what RNA is transcribed by what polymerase in eukaryotes. And that's definitely really helpful when you're just studying for the MCAT or biology classes in general. So a second question. it seems like everyone has a good understanding of this one as well. And in prokaryotes, the initial amino acid is N-formyl methionine, and it is methionine in eukaryotes. However, both are coded by the same start codon AUG. And a final question for this section. So this one is actually D, and that can be seen here. Translation actually requires GTP at every single step of the process for initiation, elongation, and termination. So during elongation, GTP actually helps bind the helps bind the amino acid to the mRNA at the A site. And during elongation, GTP helps the hydrolysis of the polypeptide from the tRNA occur, and also allows the ribosome mRNA and release factor to disassociate. I hope that clears up any confusion. So I do want to move on to mutation, just to cover what happens if you mess with this process. So if you haven't noticed, there are only 20 amino acids that humans and most eukaryotes use in their bodies, I guess. And each of these amino acids are coded with a codon, which includes 3 positions and 4 nucleotides in each of those positions. And that allows for 64 combinations. The 64 combinations coding for 20 amino acids means that there is degeneracy, which means that there are multiple codons that code for amino acids for a single amino acid. And what that does is it protects against mistakes. And this is seen in the wobble hypothesis, which is a protection against mutation. What it states is that there are several different codons that can code for one amino acid because it prevents the effect of a mutation. And this occurs in the last position of the codon on an mRNA or the first position of the anticodon on a tRNA. And if you mutate, let's say you have CUC, which corresponds to leucine. And if you mutated that to AU, it would still correspond to leucine. And that prevents mutations from actually having a negative effect on the protein product and keeps it silent, or which we'll go over later. So why is tryptophan not involved? That's a good question. I do not have the answer, unfortunately, but tryptophan and methionine are the two amino acids that don't have several codons. Methionine is coded by AUG, and tryptophan is coded by UGG. And if there were to be a mutation in either of those codons, there would most definitely be at least a missense mutation, which I will cover now. So there are several types of mutations that can occur in the DNA, and I'm just going to cover the basic ones here because the larger ones, such as insertions and deletions or copy number variants, have massive effects on genes, and there isn't too much of a point to explaining what they do besides destroying the gene product or duplicating the gene. So there are point mutations, which are specific mutations at a single place in the DNA, and these mutations are expressed. A silent mutation means that the mutation is suppressed by the degeneracy in the DNA code, and that can be seen here, where even when the C turns to a T, it still codes for the right protein alanine. There are also missense mutations, which substitute one amino acid for another, and this generally occurs when you have a mutation in the first or second bases of a codon, and what that does is it changes one amino acid for another, and you can see in the third codon, the T has been changed to a G, and that changes the amino acid product from cysteine to arginine. Now, the final point mutation is a very deleterious mutation, and it is a nonsense mutation, which creates a premature stop codon, cutting off the protein much shorter than its normal product, which generally has negative effects on the protein in the cell. And this is also known as a truncation mutation. And you can see this happen when a codon is mutated to one of the stop codons, such as TGC being mutated to TGA here. And when that codes for a stop codon, all of the codons after it are disregarded, meaning that you have a much smaller protein product. Now, other than point mutations, there are also frame shift mutations, and their effect depends on the location of the mutation. But on the average, frame shift mutations are generally have a stronger effect than point mutations. And what frame shift mutations are can be seen here. You either delete or insert an extra base pair within the DNA, and that causes that to be transcribed into the mRNA and then translated into the protein. In this case, you can see that creating a deletion here changes all the proteins after, and inserting an extra base pair here also has the same effect. But you can also negate the effect of a frame shift by having an insertion and a deletion of the same amount of base pairs normally. In this case, that did not happen because it occurred in the first location of the codon. Any questions so far? And to further explain frame shift mutations, there is a reading frame, which means the location where we start dividing mRNA up into codons. So in this case, the reading frame would start with the AUG, as it normally does, and we could shift it forward and start with a UGG. And it determines, the reading frame for translation determines the protein product. If a reading frame is incorrectly read, it could lead to a completely different protein product. So now after mutation, we have regulation of gene expression. So I mentioned an operon earlier, and I'll explain that now. An operon is a unit of linked genes that are transcribed together in prokaryotes. And we can see what that looks like using the Jacob-Monod model, which includes structural genes that represent the protein of interest when transcribed and translated, an operator, which isn't transcribed, but that is what regulates the expression of the gene, a promoter, which, like always, is the location of RNA polymerase binding, and a regulator gene, which codes the repressor that can bind to the operator. Now, there are two sorts of operons, inducible and repressible systems, and I'll get into those now. So the TRP operon is a repressible system, which means it's normally active. And this operon is in prokaryotes for tryptophan creation, but it's energetically expensive. Normally, what happens is the regulator gene, which codes for the repressor, is normally inactive, and it is activated by binding to the co-repressor. So the repressor generally doesn't bind to the operator to prevent transcription. And when tryptophan is available in the environment, it binds to the repressor, which then creates a conformational change in the repressor that allows it to bind to the operator. And what the repressor does is it blocks the section of DNA that the RNA polymerase has to pass in order to transcribe genes. And usually with repressible systems, the co-repressor is often a product of the system, and that allows for negative feedback. In this case, the TRP operon creates tryptophan. And so when tryptophan is available in the environment, prokaryotes don't want to create more because it is energetically expensive to create that tryptophan. So in this case, when tryptophan is available, it binds the repressor, which blocks the gene from being transcribed. And this is called negative control, when a repressor binding to the gene prevents transcription. On the other hand, we have the lac operon, which is for lactase creation, which is also an energetically expensive process. In this case, however, it is an inducible system, which means that it is normally inactive, and the repressor is bound because of that. The body prefers to metabolize glucose instead of lactose because it is more energetically favorable to metabolize glucose. And so lac operons are inducible in that they only turn on when there isn't enough glucose and there is lactose available for metabolism. In this case, a reactant in the pathway, or the product of the pathway, lactase, has an isomer called allolactose that is the inducer. So when lactose is available for digestion, its isomer binds to the repressor, which creates a conformational change that removes it from the operator. And that allows RNA polymerase to bind to the promoter and start transcription. However, this is also regulated by CAP, which is catabolite activator protein. And that shows positive control of this system because CAP is regulated through the levels of glucose. When glucose is low, cyclic AMP concentration increases, which activates CAP. And when CAP is activated, it also binds to its site within the lac operon and promotes the binding of RNA polymerase. for transcription of these genes. LAC and TRP operons are located in the genes for bacteria. So yes, they will be in the DNA in the nucleus. Operons are just a system for a model of genes that work together. No problem. So I just want you guys to remember that LAC operon is an inducible system, which means it's normally inactive, and the presence of a metabolite deactivates the repressor and allows transcription of the enzymes to digest the lactose. And for the TRP operon, it's a repressible system because tryptophan is necessary. So a lack of tryptophan in the environment would mean that creation of tryptophan is necessary. So that's negative control, in which the product causes repressor binding to the gene to prevent transcription. So there is an additional system for regulation in eukaryotes specifically, and this generally happens at the at the beginning of the process and transcription. So we have transcription factors, which I mentioned towards the very beginning of the review. And these transcription factors have two domains: a DNA binding domain that binds to a specific part of the promoter, or a DNA response element like the enhancer, which we'll get to later. The other domain binds proteins like RNA polymerase or other transcription factors that just amplify the effect and promote transcription by recruiting RNA polymerase. Histone acetylase is also another regulatory protein that I'll cover in the next couple of slides. Now, we have transcription factors that bind to DNA response elements, and enhancers are groups of DNA response elements where different transcription factors, different levels of different transcription factors can allow a multifaceted control of the level of transcription for the gene. The reason these groups are called enhancers is because their distance to the promoter, and they can be either upstream or downstream, forces a hairpin loop to have a proper effect, as you can see in the picture here. These enhancers can be located as far as 1,000 base pairs upstream, or even be located in an intron within the gene. There are some enhancers that bind, there are some enhancers like cyclic AMP response element, and this binds cyclic AMP to provide a positive response. There is a glucocorticoid receptor, which binds cortisol to enhance transcription, and there's a estrogen receptor, which also binds estrogen to enhance transcription. And regulation of these genes can also be upregulated through duplication of these genes. And all that does is it allows transcription and translation at multiple locations with the same gene, and that increases the gene product. Now, the final level of regulation allowed by eukaryotes is chromatin modifications. And to explain that, we'll need to understand what heterochromatin and euchromatin are. And heterochromatin is a tightly coiled version of chromatin where the genes are relatively inactive because of the lack of access to the nucleotides because of how tightly they are coiled. Euchromatin is loosely coiled DNA, and that allows for relatively active gene transcription because of how easy the DNA in those areas are to access. Now, this can be changed by histone acetylation, where lysine residues on the tail of histone proteins can be acetylated, which decreases the positive charge on lysine, and the interactions between the positive lysine and the negative DNA decrease and allow the chromatin to open. And the opposite of this can be done by histone deacetylases, which removes acetyl groups from lysine, allowing a stronger positive charge on histones, forcing them to bind more tightly and close chromatin. DNA methylation can also occur, not at the histones, but at the DNA sites. And these DNA are methylated at cytosines and guanines. And what that does is it silences gene expression by preventing access to those nucleotides with the addition of a methyl group. And this process is extremely important in development because the right genes need to be turned on and turned off at the right times to promote proper development. And finally, I have 3 more questions for you guys, and that's pretty much it. So like always, just type it in chat or let me know when you guys have the answer. And I do want to tell you guys that this one may take a little bit to do. Sorry, I did realize I actually meant to put coding strand instead of non-coding strand on here. So I apologize for mixing that up earlier, but yeah, it is coding strand. And if anyone would like me go over this one step by step, I can definitely do that. Just let me know. If not, I guess we can move on. But the answer is D, methionine. And that is because a coding strand reading 5' to 3' CAT would mean a template strand, or the sense strand, meaning showing a 5' to 3' of ATG. And when you translate that to mRNA, it would show up 5' to 3' as AUG. And when you look up the mRNA code for AUG, it is the only codon that corresponds to methionine. So here is the second question. Great, this one is negative control. And that's because when a activated repressor binding to the operon prevents transcription, that's negative control. And then the final question I have for you guys tonight. So the right answer for this one is B. It is unlike histone acetylation in that it silences gene transcription, and methylation does that by preventing access of polymerase to DNA by binding those sites with a methyl group, while histone acetylation loosens histones, allowing greater access to the DNA. So those are the last of the content slides that I have for you tonight. I do want to mention, I do work with MCAT 101, and any donations that you guys have would help. We're super close to our goal of becoming and registering as a nonprofit. So please donate if you can. And please send us feedback or ask me or MCAT 101 questions, and send me your email so I can add you to the list for the newsletter and topic sessions. Thank you guys for being here today. And if you have any further questions, I can help.
Hey, do you mind going over that real quick about how to tell the coding versus non-coding and the with the DNA strand?
Yeah, definitely. I can do that. 1 second. Let me find the right slide for you.
I just find that kind of tricky because it's like I don't know to look for certain sequences or is it
Gotcha.
just based on the directionality of it or
Yeah. 1 second. Let me write this down so I don't confuse myself as well. Because there's 3 different names for it, right? There's antisense, coding, and template.
right?
So
Just like coding and non-coding and
exactly.
right.
So I do want to mention that this has to do with a specific mRNA or protein, right? The only way, the only time we can give a name to the separate strands is when we're looking at a specific protein and where it's transcribed from. Does that make sense?
Right.
So when we find a specific protein, and let's say that it's being transcribed as it is in the top picture, we know the DNA strand that's being transcribed from will be called the antisense strand, or the coding strand, or the non-template strand.
Okay.
So
Okay.
those 3 names are the same. Antisense, coding, and non-template are the same thing.
Gotcha.
And on the other hand, the sense strand, the non-coding strand, and the template strand are the same thing.
Okay.
And those names correspond to the strand that looks most like the RNA transcript. So if you look at the bottom strand and you look at the RNA transcript, they look extremely similar except for U's being in the place of T's, correct?
Correct.
And all that means is that the RNA transcript is being based off the template of the template strand, right?
Right. Now, is that the top strand, the template strand, right?
The bottom strand is the template
the
strand.
bottom. Okay.
Yeah,
Okay.
the one that looks most like the RNA transcript is the template strand, because
Gotcha.
that's what the RNA transcript is based off of.
Okay. Okay. Would it in a question stem, would they just give us both strands or would it be set up, presented something like this kind of or?
Well, if they're going to determine, if they're going to ask you to determine anything to do with the sense or the antisense strands, they're going to have to give you a specific mRNA or protein product, and they're going to have to show you which strand it's. They're going to have to either show you which strand it's being transcribed from, or they're going to ask you to compare the sequences, right?
Right.
So the sequences that are the most similar with the RNA means that it is the template strand or the sense strand, the one that makes sense, right?
Right.
Yeah,
Mhm.
that's the way you're going to have to determine it.
Okay. Okay. And then the other strand, we just, it just does regular base pairing, right, with the RNA, the antisense strand.
Yeah, the antisense strand binds to the RNA just to give it the complementary sequence so that it can recreate the template strand.
Okay. Okay. All right. Are we
Yeah.
dealing with like items that know that, like, how does the enzyme and everything know to like match up with everything? Do you see what I'm saying? Is this RNA polymerase doing that? You see what I'm saying?
To match up with everything, as in
trying to undergo like translation, basically. Like how does it know what sequence to pull apart? Do you see what I'm saying? Like, does it scan the DNA some type of way to know, like, where helicase comes in, it unwinds the DNA, unzips it. But then how does it know where to begin? You know what I mean? This process, is it a sequence we look at or
so
not
yes,
really?
that is determined by the transcription factors and the enhancers. So basically, when you want to induce transcription of a particular gene, you're going to see a greater concentration of the transcription factors that correspond to that gene and an activation of the enhancer that corresponds to that gene. That allows an opening of the DNA, or it tells the cell, this is the gene we want to transcribe right now because of some stimulus. And so helicase and topoisomerase know to open up that specific area, and RNA polymerase gets recruited by those transcription factors.
Okay, okay. Does that have anything to do with like those palindromic sequences and all that? You know what I mean? Like tandem repeats, or you know what I mean? Like it knows about a specific sequence once it scans, you know, to pull it apart. You know, does it have anything?
I'm not 100% sure. I can't say off the top of my head if it does scan for any tandem repeats. I
Right.
do believe that it is recruited and told where to go by transcription factors and enhancers.
Okay. Okay, gotcha. Okay. And my last question for you is like when the nucleotides bind, does that come from the OH group and the phosphodiester bonds, or
Which nucleotides bind?
like I'm talking about like in general with when RNA polymerase adds new nucleotides to the growing DNA strand, what's the chemistry like going on there? Like with the OH group and the phosphate group? Like
when
how does
you're
that work?
in the transcription of the RNA.
Right.
So the RNA basically pulls free RNA, like nucleotides, from within the nucleus and gives them a place to form a bond with the sugar phosphate backbone of the preceding RNA molecule.
Okay, so it's the phosphate groups that are forming the bond, kind of.
Yes, it should be the same as DNA.
Right.
When we look at let me pull up that slide.
Mhm.
I believe, yes, it should be just like this with the change of thymine for a uracil, and the
Okay.
hydroxyl group being, because the hydroxyl group that does the bonding isn't the one that's missing, if that makes sense.
Right. Okay.
So the bonding is the exact same as it is in DNA.
Gotcha. So if let's say for instance, if we wanted to add like a nucleotide, another, like adenine or something, where would it add at?
It would add to the 3' end of the existing RNA transcript.
Gotcha. Okay. And then it would give off water?
Yeah.
Okay.
It's the exact same.
Gotcha. Okay. And then it always goes in that 5' to 3' direction.
Yep.
But it has
the only time they're synthesized is in the 5' to 3' direction.
Gotcha. Awesome. Okay, thanks a lot. Really appreciate your help.
No problem. If that's all from everyone, I believe this is the end of the session. Thank you guys so much for coming. Please consider donating. I will be posting these slides up, so thank you guys.